All right, this is part 2 of chapter 12 on the cell cycle. Now we're getting into aspects of regulation of the cell cycle. So every cell divides at a different speed, different times for different reasons. There's, of course, programming signals that determine when that is. So the frequency of cell division varies with the type of cell, and there's regulation aspects at the molecular level that turn the cell cycle on or off. So specific molecules that are in the cytoplasm regulate the progress. So here's a experiment where they took mammalian cells that they cultured at 2 different phases of the cell cycle. And they were triggered to fuse. So here we see a cell in the S phase of interphase, and here's a cell in the G1 phase. When they combine them, both of the cells that were fused entered the S phase, even though this one was in the G1 phase. So there was something in the cell of the S cell that triggered the G1 cell to continue the cell cycle. And of course, the same goes for this in experiment 2. Fusing 2 cells at different phases of the cell cycle suggests that molecules present in the cytoplasm of cells control the progression of phases. So here's the molecular clock that's very typically shown, and we'll tie to our lab G1, S, and G2. Here's M and cytokinesis. Now, at every spot within G1, and then these other trigger points, there's these checkpoints, which are very important points at which the cell determines whether it's going to continue through the cell cycle or whether it's going to stop. G1 checkpoint, there's actually also a checkpoint between S and G2, but that's minor. Here's a G2 checkpoint right at the end of G2, and then finally an M checkpoint following mitosis there. So there's, of course, green light, red light, signals that say go or stop, and you'll see later that cancer is when they're all green lights. Now, the regulatory proteins that are involved in this control we group and call cyclins and cyclin-dependent kinases. Now, you already know previously that kinases phosphorylate things and thus activate them. So cyclin-dependent kinases we'll call CDKs from now on. And cyclins, as you know, are proteins that are associated with the cell cycle. Now, here we have the activity of cyclins and CDKs, and you can see how they fluctuate during the cell cycle. MPF is a CDK. And here we see cyclin in the, I guess this would be purple and this would be violet. I don't know the difference. But you can see cyclin activity triggering or starting early in interphase, whereas only in mitosis do you have the CDK activity. So synthesis of cyclins begins in late S through G2 because the cyclin is protected from degradation. So it's allowed to accumulate. It's not being used up yet. And then the cyclin molecules combine with the CDK molecules to produce MPF, okay, and thus allow, when in the presence of MPF, we can pass the G2 checkpoint and begin. So MPF essentially triggers that, promotes mitosis, phosphorylates things, and activates them. And then MPF gets degraded, which says essentially stop. Here's the degraded cyclin. And then we have G1 that continues and then will stop at the G1 checkpoint. So there's these are just examples of a few of the cell cycle checkpoints and/or molecules that we know that monitor the cell cycle. There's other things that we call growth factors that stimulate cells to divide. So here we have an experiment where we have connective tissue that gets cut up, and you degrade the ECM, the extracellular matrix, and you have free fibroblast cells, which are early cells in development, or even adults have them, but they're early on cells that will become connective tissue cells, fibrous cells. Anything that ends in a blast, just so you know, is a precursor cell. So myoblasts are precursors for muscles. Myo means muscles. Now, here they took those cells and they grew them out, and they grew them with PDGF and without. And then basically they incubated them. You can see here the difference. And what they found was that there was density-dependent inhibition. So when there was a lot of other cells in the way, the cells stopped dividing, density-dependent. And then most animal cells also exhibit anchorage dependence, meaning they must be attached to something to divide. If they're just floating around in solution, they actually won't divide. It's not entirely clear why that is, but it is known that that's the case. Then we have cancer cells here specifically that for some reason they are not density dependent and they're not anchorage dependent. So they can be floating around in your bloodstream and they can divide, or they cannot be attached within a within a tissue and still divide. They don't exhibit this inhibition, and it's not entirely clear why. They can just keep forming and keep dividing. So they don't abide by the normal control mechanisms, and as a result, they can potentially form tumors, both benign and malignant. Now, malignant tumors can invade surrounding tissues. We call this the ability to metastasize or move through the bloodstream, where secondary tumors will form. So if you have, for example, lung cancer, and that lung cancer metastasizes to the lymph nodes, you now have the same kind of cancer in two places. You have lung cancer on the lungs, and you have lung cancer on the lymph nodes. And they will attempt to identify the original location of the cancer and treat it based on that original cancer type. And so you can see that here. Here's a metastatic tumor entering into the bloodstream there. And we're done.